Okay, everybody, I think we can get started. Again, this is Neil Andrews, editor of Pain Research Forum. I'd like to welcome you to today's webinar on an introduction to pain neuroimaging in humans to be presented by Dr. Lauren Atlas. In terms of our format today, it'll be our usual format. Dr. Atlas will present a talk for about 35 to 40 minutes, and then the remaining time will be devoted to audience questions and discussions. Speaking of which, we hope everyone will submit your questions. You can do that at any time during the talk, after the talk, at any time by typing them into the Q&A area of your Zoom window. We also encourage everyone to join the conversation about the webinar if you'd like on Twitter and do some tweeting using the hashtag PRF webinar. And the webinar is also streaming live on the PRF Facebook page. And so just a shout out and welcome to everyone on Facebook who is listening to the webinar that way. So now I'd like to introduce our moderator, Dr. Petra Schweinhart. Petra is the head of research in the Department of Chiropractic Medicine at Balgrist University Hospital and the University of Zurich in Switzerland. And Petra is also a PRF editorial board member, and we're really thrilled to have her here today to moderate the webinar. So Petra, welcome. Please take it away.
Yeah, thank you, Neil. So it's really my pleasure to introduce Lauren Atlas to you. She's a tenure-track clinical investigator at the National Institutes of Health. She's the chief of the section on effective neuroscience and pain in the National Center for Complementary and Integrative Health and holds joint appointments with the National Institute of Mental Health and the National Institute on Drug Abuse NIDA. Her laboratory integrates experimental psychology, neuroimaging, psychophysiology, and computational approaches to understand how expectations and other cognitive and affective factors influence pain, emotional experience, and clinical outcomes. Her current projects focus on dissociating components of expectancy, relating pain with other types of affective responses, and understanding social influences on pain. So that makes sense then that her long-term goals include revealing how specific features of the clinical context and interpersonal aspects influence patient outcomes, as well as determining whether expectancy-based processing is altered in specific patient populations, including those with pain. Dr. Atlas received her PhD in psychology from Columbia University and completed postdoctoral training at New York University. So it's really our pleasure to have her speaking to us today.
Thank you so much, Petra, and thank you, Neil, for inviting me to do this webinar. I'm really excited to have the opportunity to give this overview and kind of introduction to a primer that my colleagues and I recently wrote on pain neuroimaging in humans. So I'll go through my talk for the next 35 to 40 minutes, and then I'm excited to hear what kinds of questions people have about fMRI. So this talk is really geared after, based on the primer that Masi Moyedi and Tim Solomons and I published last year in the Journal of Pain. And that primer kind of came out of a discussion that I think we have often, which was debating about what needs to be reported in an fMRI study. So specifically, for pain researchers, if people are dropping out during the study, do we actually report that in the methods? How do we report it? What kinds of things actually go into a paper? And we started with talking about that, but then realized there are many different kinds of decisions that one needs to make in order to run an fMRI study or report it or read one or make a decision as a reviewer. And so we thought that it was especially in light of all the interest in brain imaging for pain, now that we have the opioid crisis and new funding opportunities and many reasons to be paying attention to pain, that we could kind of introduce a critical view of how to conduct an fMRI study and what pain researchers specifically should be paying attention to. So I definitely want to acknowledge Masi and Tim's contribution to this work, which I'll be talking about today. So the number of brain imaging studies has, and specifically those using fMRI, has exploded over the 20 or so years since the first fMRI study of pain. So we see this exponential growth that's continuing to increase. And so we think that this is obviously a tool many people in our field are choosing to use. And we need to make sure that people understand what goes into running a study and how to critically evaluate it. So just to give the briefest of overviews on pain fMRI, just to give you kind of a sense of where we've come from and where we are now. The first fMRI study of pain that was published following huge breakthroughs using PET imaging to show brain responses to pain in humans. Jaron Davis showed that electrical nerve stimulation elicits responses in somatosensory cortex that track pain and are specific to pain. So here's activity over time within this S1 region. And by 2011, there had been 132 different studies using PET or fMRI to measure brain responses to pain in patients and healthy volunteers. And today we have studies that look more like this, where we're looking at connections across regions. So this is a recent study from Etienne Vachon-Presseau looking at predictors of placebo analgesia. So kind of my goal today is to talk about how we can use this crazy huge machine to generate images like this. And I really want to kind of walk you through the steps of decisions that people make so that you can be a critical connoisseur or an imager if you are a grad student who's planning to get involved. So the goals for my talk today are to help you understand the constraints and limitations for using fMRI to study pain, to help you learn which elements should be addressed in any fMRI study. So if you're a reviewer and you're reviewing an fMRI study, our primer and this talk give you a sense of things that should be addressed. And if they're not addressed, you should feel free to ask the authors about these elements, to give you an introduction to the statistical approaches we use to conduct fMRI analyses. And this is just a brief introduction. There's much, much more to say, but I'll be providing resources for you, which you'll receive after the seminar. fMRI researchers who are newly getting involved should be able to either critique or defend choices for analysis. And hopefully everybody will be both skeptical and a connoisseur of pain neuroimaging. So those are my goals. The things that unfortunately I'm not going to be able to do within the next 35 minutes is give a history of pain neuroimaging. That I've done 40 minutes talks on that, and even that is kind of tight. So I had to make a decision. We're really just going to be focusing on the methods today. I won't be talking about brain imaging other than fMRI, and I will not be addressing most of the exciting debates in our field, things like the validity of using fMRI as a biomarker for pain, the utility of acute pain as a way to understand chronic pain, things like social pain, whether or not that exists, hashtag cingulate gate, differences between pain and salience. I think these are all like super exciting. It's why I work in this field. But I think my goal is for you to have the tools to read the papers in these areas and be discerning in terms of whether that study is actually asking that question. So with that, the talk is structured kind of the same as an fMRI study would be if it were reported, and that's similar again to our primer. So I'll begin with a brief introduction to fMRI. Then I'll turn to fMRI methods and results. We'll talk about issues concerning the fMRI environment, what people should be reporting, how to optimize fMRI study design. I'll give you an overview of different types of fMRI analyses, from data acquisition to group level results, and briefly address some of the new approaches, although unfortunately I don't think I'll have time to go into that in detail. Finally, I'll talk about some key issues in interpretation and inference from fMRI studies. So functional magnetic resonance imaging, I'm sure many people have heard, maybe you volunteered for a study or you're aware, but an MRI scanner is a big magnet. So it has different magnets embedded into the scanner itself, as well as the gradient coil. And what happens is there's an overall magnetic field, B naught, and the gradient coil basically sends different types of excitation pulses that are sent at specific intervals, so often like every 2 seconds, and we use that to get a measure of brain activity. The first study, or first images of ongoing brain activity, recently I saw this through MGH, Ken Kwong showed visual stimuli to an individual. And so here you can see this is visual cortex, and it's lighting up, basically, the activity that is reconstructed from the MRI scanner is associated with changes in the stimulus. And so here is a paper that came out about at the same time by Ogawa in Michigan, I think, in PNAS, showing that when light is displayed to a subject in the scanner, you see these increases in signal intensity in the visual cortex, part of the occipital lobe. And so this is the first time you see these colorful blobs. They're just a measure of signal intensity here. Later, you'll see how currently the blobs are a measure of statistical strength. And so the reason that we get these images is because the idea is that when the synapse fires and a neuron uses energy, it needs to convert glutamate to glucose, which requires oxygen. And so there becomes a difference between oxygenated hemoglobin and deoxygenated hemoglobin, and those turn out to have differences in terms of their magnetic spin properties. So basically what happens is when we use this B1 signal to excite the response, it kind of like sends all the magnetic pulses in different directions, and then the receiving coil waits until how long it takes for the signals to get back in line. And so given that there are differences between oxygenated and deoxygenated hemoglobin in terms of their magnetic properties, that's why the MRI scanner is able to pick up on differences in activity. So we don't actually know the difference between the relationship between this magnetic property and neuronal firing itself, but we do know that this kind of change in blood flow, oxygenated blood flow, elicits a difference in this magnetic signal, and that's what we call BOLD signal, blood oxygen level dependent signal. So you can, I'm going to send links, and you can tune into talks about fMRI imaging specifically and the physics behind all of this. So fMRI, when compared to other methods, the reason it's become so popular for human imaging is because it has a great balance between the temporal and spatial resolution. And this is getting better and better now that we have high field strength. So previously, most studies were about, it took about 2 seconds to image the entire brain, and now people can image the entire brain on the order of half a second in some scanners, depending on what part of the brain you're looking at, how much you're looking at. And similarly, voxels, which are the 3-dimensional pixels that we use as units of the brain in brain imaging, previously were about 4 millimeters and now can be less than 1 millimeter cubic measurement. So we're getting better and better at improving both the spatial and temporal resolution of fMRI. And that's really been the big draw in terms of using fMRI to measure pain, because we can look at the response to a stimulus that we know, even if it's very brief, we can look at the brain response to that stimulus. So after the seminar, we will be sending you a list of resources for fMRI. These are really to go beyond my talk and our primer and consult with statisticians and methods experts in the field. So some of the tools that I really highly recommend are this thing called the Cobitas report. It's a group that kind of made the best practices in data analysis. And they have a checklist for kind of what steps to go through for every fMRI study. And we highly recommend that people use that when you're reporting your results. Tori Wager and Martin Lindquist have a 2-part Coursera class on the principles of fMRI that goes into depth with the types of issues we'll be addressing today. Jeanette Mumford, who's an fMRI statistician, has an awesome YouTube page with tons and tons of videos on every different aspect of fMRI analysis and statistics. The NIH, where I am, we have a summer course every year with 3 classes a week dedicated to fMRI, and all of the previous lectures are archived online, and you can also tune in this year and get them as they come. And finally, the Organization for Human Brain Mapping, which is a society that's really dedicated to improving fMRI methods, has recently released this online library with all of the lectures from their conferences going back to 2013. And every year there's a pre-conference workshop day, which includes courses on things like advanced fMRI analysis, structural imaging, computational imaging, machine learning, many, many, many different topics. So I think one of the take-homes to me is that we as pain imagers really can take advantage of the huge insights being made by people who are working on fMRI specifically, and then we can think about how to adapt them to our needs. So how do we kind of get started? How do we go from this constrained environment with a subject lying in this large magnet to getting some kind of insights on this kind of acute pain in the environment or chronic pain in individuals? So I'm going to be walking through kind of each of these aspects and where I think there are important questions to be aware of. So first of all, by doing a study inside the MRI scanner, we have huge constraints on the type of samples we can actually collect data from. So there's exclusionary criteria for participants. First of all, this is a really tiny magnet. So there are important size limits due to the size of the scanner bore, as well as the head coil. So you can see this is, your face goes in this tiny little thing. And so we've had participants who don't fit in there. And with patients, you know, that can be, with different types of patients, that can be an issue, talking to them about whether they're gonna fit or not. And we know that that individuals with obesity have higher rates of pain. So it may be difficult to even study those individuals using fMRI. A second issue is that because it is a big magnet, individuals can't have any kind of ferromagnetic metal in order to be safe in the scanner. So they cannot go in there with a pacemaker. Patients with maybe phantom limb pain who have devices might not be able to go in the scanner if they're not MR compatible. Elderly patients who have pain might be more likely to have devices. So this also is another exclusion, and adolescents with braces that came up on Twitter. So not everybody's going to be able to be in the MRI scanner because of safety constraints. And finally, you have to lie in the scanner from anywhere between, let's say, 20 minutes at the least to an hour and a half at the most. And so that can be claustrophobic for anybody, whether or not they have any other comorbid conditions, but also it's highly uncomfortable. And so pain patients, maybe patients with low back pain or other types of pain conditions, might find it particularly difficult to lie still in an MRI scanner over time. And we'll talk about motion in a little bit. So what this has done to our field has left us with an over-reliance on convenience samples, often healthy undergrads, doing acute pain tasks. And so when we are reviewing these studies, we really need to ask, is this a population from which the authors can make these conclusions? One thing that researchers who are interested in specific populations might do is run the same study outside the scanner as well and show that the behavior isn't modulated by the scan environment, and that if you draw from the wider population, you still see similar effects, at least on behavioral outcome measures, although you won't be able to look at brain imaging. But so these kinds of constraints really kind of are the first step to severely limiting what we can do and what we can ask using fMRI. A second huge issue is the constraints that affect the experiments themselves. So once again, it is a big magnet, and this means that we can only use MRI compatible devices that are not affected by the magnetic field, that don't have any metal components. And so for that reason, we have many fMRI studies that administer acute thermal pain, as well as electric shock, and some other people are getting pressure devices that are able to be administered in the scanner, or laser, but that's very few. So we have this overabundance of reliance on specific kind of stimulus delivery equipment, and that prevents us from making the same kinds of insights or even doing comparisons of brain activity between, let's say, thermal pain and cold pain or cold allodynia, chemical pain, tonic pain. We'll talk a little bit later about why these kinds of longer lasting pain stimuli aren't as easily suited to the scanner. And even within MRI compatible devices, they can differ depending on the type of scanner. So we're not going to talk about it very much, but MRIs differ based on field strength. And so not every equipment that's compatible with a low magnetic field is going to be compatible at a higher field. Secondly, the subject is lying in the MRI scanner while they're doing your task in most cases. And so you have to create a very artificial way to have the subject experience stimuli. So one typical setup is that there will be a mirror in front of the subject's face and a projector that bounces an image off of the mirror onto in front of the subject so they can make responses. They need to make responses using specialized devices that are compatible with the MRI, so you need to kind of buy specialized equipment in order to get responses in the scanner. The scanner is really loud while it's on, so people can't just tell you their pain rating. And finally, you need to, we'll come back to this when we get into the analysis section, but you need to know the timing of the events. So you need to carefully lock the onsets of your stimuli with the MRI scan time. And usually we do that with kind of specialized computer programs that synchronize the stimulus delivery and the scan onset. But these are all things that one needs to think about before actually going ahead with an fMRI study. And if you don't have a way of inferring the time the stimuli were delivered, it can be much more difficult to do fMRI analysis. Now, the third huge experimental constraint is the issue of head motion. So one reason this is an issue, let's take these headbangers here. If they were in the fMRI scanner listening to heavy metal and banging their heads, we're not going to be looking at the same brain region over time. So we won't know if we're looking at the prefrontal cortex or the anterior cingulate or visual cortex if their head is moving, because the MRI scanner is just measuring from one location in space. And so if your head moves and you're looking at the same spot, you don't actually know what you're looking at. So that would be a simple constraint. And to avoid that, we basically put lots of pillows in with the head and try to help the person not move. And people are building new ways to do that with devices that are actually like shaped specifically to a person's face. But movement actually has many issues even beyond potentially making us unsure what region we're looking at. So here, I took a video of activity in different voxels of the brain using AFNI. So this is really nice. You can see activity over time. And in this voxel right here, what I'm seeing is this big jump in activity at that period. So what I'm going to do to inspect that is I'm going to trace along time on the x-axis, and you can watch this brain here as we get closer to that big jump. And what you see is that that was actually head motion. But what happens when the person moves their head is it also introduces an artifact. So I don't know if you saw that stripiness, but basically motion within the MRI environment itself creates artifacts and perturbs the signal in the MRI scanner. So when somebody moves, it's not just a question of which voxel you're looking at, this one or this one, but also the signal itself becomes decayed, and that affects the whole brain. And so this basically introduces another restriction, because we have to choose subjects who can lie still in the scanner. So this might be more difficult for people, let's say younger individuals, like if we're studying pain in children, or it'll constrain our ability to study things like movement-induced pain, because we can't ask people to move in this scanner. So, you know, we're trying to look at pain, which is some things that people want to escape if they can, and yet it's ultimately so critical to have people remain completely still. And so what we do, assuming that people don't move too much, we'll talk about what too much is in a second, we do what's called motion correction. And so what we do is we kind of like align, even though, since there's small movements, so I'm not going to replay the video, but the brain was moving, you know, very, very, in very small steps. So this is the 6 different dimensions, basically in space, that the subject moved in that block. And so you can see there's these small movements over time. And so what we do in a step called motion correction is align the brain into the same space for every volume of time. So every 2 seconds we get a picture of the brain, and then basically during motion correction we line up those pictures with of the brain with themselves, so that when we do our analyses we're looking at the same brain region. The voxel corresponds to the same brain region over time. But because I showed you the types of artifacts that are introduced, even if we line up the brain so that it looks like it's in the same space, we're still left with potential effects on the MRI signal itself. So when we do statistical analyses, we have to include motion as a nuisance covariate in our analysis and make sure that our activity that we look at later when we're interested in our the functions of interest is not attributed to motion. Finally, there are ways to actually remove periods of large motion. That's called scrubbing. Or if a subject exceeds a certain motion threshold, often something about like 2 millimeters, if you've got a 3 or 4 millimeter voxel. So if somebody moves more than 2 millimeters, you might exclude them from analyses altogether. And again, I really think people should consult the references to like our primer, as well as the references I'll send, to get much more information on how to make these kinds of decisions. Finally, most people who have an fMRI scanner at their institution might have recommendations from the local MR physicist about what imaging sequence to use to collect fMRI data. But the selection of sequences actually may depend critically on your questions. So here are different examples. So this is actually the exact same time point. This is using what's called multi-echo data. And so this is different durations of this excitation threshold. So 17 milliseconds, 22, and 33 milliseconds. Oops, sorry. And so what you can see is that the region of the orbitofrontal cortex here drops out as we move to a larger TE, but we get better contrast resolution. And so we have ways to combine across these different echo times and get what's called, in this case, optimal combination. But one has to really make decisions about, you know, you don't always have this multi-echo sequence, but also even in doing optimal combination, there are strengths and limitations. So here what you can see is that in order to get adequate coverage of the regions I cared about down here, I had to cut off the back of the brain. And so we have to make decisions about what size slices we prescribe. So the imaging is actually a big block of slices, and we collect brain data 1 slice at a time. And so in this study, I will not be able to make any inference about what's happening in S1 because I don't have S1 coverage. So sometimes you have trade-offs between what brain regions you want to make sure you can recover signal in, if they're sensitive to dropout, and you lose other regions in turn. Now this also becomes even more intriguing as you move to trying to get inferences on things like midbrain structures or even brainstem imaging. And brainstem imaging requires not only special sequences, but also special head coils, head and neck coil, and special sequences. So we can't always use fMRI to get inferences on every brain region. We have to think before we even start the study about what regions we care about. And so overall, just to kind of make sure I get through all the topics, in terms of the materials and procedures that should be reported, people should report all of their decisions in these areas. All the details of your study design, this imaging acquisition, how stimuli were selected, what subjects were told, did any subjects withdraw because of the fMRI scan environment or because of the pain stimuli. We talk about this in detail. In addition, because your decisions may depend on your regions of interest, it's really important, and I hope our field moves toward these new efforts to do open science and pre-register our hypotheses. That way we can specify at the outset what we're planning to test and design our study accordingly. And we should conduct power analyses to make sure our fMRI samples are adequate sizes. And again, we talk about this in detail in the primer. I also recommend that people check out the Cabitas report on this as well. So once you have fMRI data, you need, before you can do statistical analysis, you have to do a whole series of steps called pre-processing. And I'm not going to walk through all of the steps, unfortunately, because we don't have time. I already talked a little bit about motion correction, but there's many decisions along the way that kind of help get you from raw data out of the MRI scanner to the point that you can actually look at the relationship between the brain data and your activity. And there are many different neuroimaging analysis softwares that can be used. And unfortunately, many of these different programs use different algorithms for different steps of these processes. Fortunately, within the fMRI field, there is a new effort to try to synchronize approaches across different types of studies and different labs. And this is through what's called brain imaging data structure and fMRI prep. And this has default assumptions that draw on basically the best of each of these kinds of routines and allow us to kind of have some defaults that are agreed upon. So that is one way if somebody's getting involved. If you start out with BIDS and fMRI prep, you may not need to, you should still understand all the steps involved. And we do talk about those in depth in the primer. But this is a way to at least kind of help our field have some standard decisions and therefore be able to compare across studies. Now, one step that is important to mention, so we've been looking at the functional MRI data, but this is structural MRI data. This is a brain, a subject's brain in the original space. So this is like the space of their actual skull as imaged in the MRI scanner. And in order to look across subjects, what we need to do is what's called normalization, where we kind of use those programs I talked about to figure out how to turn this subject, how to register their brain with this template, the Montreal Neurologic Institute template. And so what you see that, in this case, AFNI did, is it kind of like bent the subject's brain in different dimensions so that it's in the same space as the template. And so when you're doing pre-processing, it's very important to make sure that you got a good registration to the template brain, because that way you can use things like atlases to ask, what is this structure right here? And also to make sure that the functional data lines up with the anatomical data, which is the step of co-registration. Once you have your data all pre-processed, then we move on to generating our statistics using subject level analyses, then group analyses, and more advanced approaches that I'll talk about briefly as well.
Oh, hi, Lauren. I'm so sorry to interrupt. I just want to remind you, we have about 10 to 12 minutes left.
That's perfect.
Okay, sorry to interrupt.
No problem. So when, oh, I'm muted?
No, you're okay.
Okay. So I mentioned at the start that when there is activation in the brain, that's really a difference between oxygenated and deoxygenated hemoglobin, which can be read by the MRI scanner. through the complex physics that I alluded to. And what this basically generates is a delayed response in the bold response, which we call the hemodynamic response function. So it's basically like the delayed blood flow response and how it looks given the MRI scanner. And so what we can do, if brain activity is responding immediately to a stimulus, there's always going to be about a 4 to 6 second delay in its response as measured by the MRI. And so what we do is basically we take the timing of the stimulus and we convolve it, basically combine it with this HRF function, and we generate an ideal time course of what it would look like for a brain region to respond to this stimulus that's presented here in what's called an event-related design. Now, it's important to acknowledge that the hemodynamic response function and its shape and delay can differ depending on the brain region you're looking at. And so oftentimes we just assume a fixed canonical HRF, but there's many different types of flexible approaches to be able to actually fit the best HRF for different regions, or even to model the HRF itself using what's called finite impulse response functions. So one thing researchers need to decide about, which we don't often talk about, is how are you modeling the HRF? Here I'm just going to show you what it looks like to use a very simple HRF model. It doesn't even have these undershoots or initial dips, but how it gets fit to brain activity. So this is activity within a voxel over time. And what you see is that this is when we applied high heat stimulation to that subject in this box. So they had 4 trials of high heat. They also received medium temperatures and low temperatures, and the heat was on for 10 seconds. So basically this is a 10-second, what's called a boxcar. So it's basically like the stimulus stays on for a little while, convolved with an HRF that doesn't have an undershoot. And we can fit that to the brain activity along with a bunch of other regressors. And this is what we get ultimately as our best fitting response, having regressed out things like motion, nuisance covariates. And so we can basically use that fitted response to get a sense of whether a brain region is responding to a given type of stimulus. And so this is actually a voxel from the dorsal posterior insula. And so what you see is it is, the fitted response at least suggests that it is really responding to high heat pain. So what we're doing there is basically standard statistical regression with just a lot of extra predictors. So, and we're doing that for every voxel in the brain. So your dependent variable in this case is the brain activity in the voxel over time. Your input variable, the X matrix, is your predicted activity based on convolving your event timing with the hemodynamic response function, as well as a bunch of nuisance covariates that I'm not showing here. So we model things like known motion, any spikes in the MRI signal, artifacts, filtering so that we have the right, we get rid of low frequency fluctuations, and we use intercepts if we stop and start the scanner because the overall scan activity level, average activity, is going to differ by run. And in the end, when you do that, you get a map of coefficients or betas for every voxel in the brain. And this is what's passed to our group analyses, right? So this is actually the statistical relationship between a regressor or a prediction and brain activity in each voxel independently. And for an individual, we can threshold this using statistics. But before we get into group analyses, I just want to talk a little bit more about setting up the first level design matrix. So as I said earlier, you need to know your stimulus timing, and you have to design your task so that you can make decisions about the timing. And you measure brain responses as a function of the thing that you manipulated in the study. So the stimulus or the condition you're manipulating. And, if it is a task that doesn't have a known timing, that can be very challenging in terms of deciding how to set up a model. We can do brief event-related fMRI or block design. And then we can use what are called parametric modulators to look at whether the activation or brain intensity scales with some kind of known behavior. For example, intensity of the stimulus, pain rating, or predictions from a computational model fit to some other outcome. So in this design matrix, this first column is every time that subjects had to make pain ratings. And then each of these columns are the different temperatures that they received. But the second column after each one is the pain rating they gave on that child. So here I can look at the overall average response to this temperature. But here I can look at how much the brain response varies as a function of the subjective rating. And so we make decisions about what to actually model, and reviewers should be able to ask people about their decisions in that step. I'm gonna skip over, these are examples of some studies. So this is an early study from Vanya Apkarian looking at modeling the stimulus versus modeling the subjective pain. And Christian Buchel modeling different types of responses in the brain and coming up with different solutions in terms of whether the cingulate is tracking stimulus intensity, pain intensity, or just knowing that the stimulus is on. So you can use this general linear model to kind of pit different types of responses against each other. And then what you do after you have each individual's first level analysis, you put them together and you just use statistics, the standard, depending on what your study question is, to run a one sample t-test, between groups t-test, look for correlations with behavior, etc. And what you get from that is, again, a statistical map with a value for every voxel about whether you had a significant difference across the group relative to 0, or whether the groups differ between them, or whether you had a significant correlation with a behavioral outcome. Because we have tens of thousands of voxels, we're running tens of thousands of statistical tests, which leads to many false positives. And so we need to kind of reduce our threshold to take into account that we're doing many tests. But rather than just kind of choosing a threshold arbitrarily, there are many ways to correct for these multiple tests. And so I'm not going to get into those, but here is kind of using this approach called Familywise error correction. This is the response in bilateral insula is really where I can be most confident that I saw differences between high and low intensity stimulation. A different way you can do this is to limit your search space to regions of interest. I'm not going to talk about these in detail because I think we described this very well in the paper, but there are many different ways to kind of identify your regions of interest, including meta-analyses. And that way you can reduce your search space to regions that you think are likely to be involved. So I think I have, like, let's say 3 more minutes. I want to go over just 3 more points. One is all of the examples I've given so far are task-based fMRI, where a stimulus is presented at known timing and you're measuring the effects of that stimulus on brain activity. But people also, for the past 20 years, have done a ton of studies using resting state fMRI. And in that case, there's no task, and you're looking at spontaneous brain activity or intrinsic connectivity. We'll talk about the term connectivity. So basically, people are just lying in the scanner, hopefully not sleeping, and you measure the correlations between brain regions over time. And this has really revolutionized the field, because it basically shows that there are reliably correlated networks, including, I think most relevant for us, is the default mode network and the salience network, both of which have been implicated in studies comparing patients and controls. But when we see connectivity or differences in these networks, it doesn't mean that there are underlying differences in the anatomy without directly using other methods to actually measure differences in the anatomy. So there's a huge literature on resting state, and will send those resources around after this. So I just think one of the most important points to make, which I think I know from colleagues of mine is something that is confusing for people, Functional connectivity is just correlations between regions over time, correlations in activity over time, but it has nothing to do necessarily to anatomic connections or circuits. So we can put a seed in a region. So here in PhD, we can just extract the time course and look for other regions that are correlated. And here we see some inverse correlations in this region of prefrontal cortex. We can do effective connectivity. There's a bunch of different methods for trying to look not just at correlations between two regions, but how one region might influence another. And recently there are approaches to measure whether the connectivity changes over time. And we recently just had an accepted review of this in pain reports. Finally, the last kind of approach is basically rather than using the brain as an outcome, we can use the brain as a predictor and use behavior as the outcome. And in this way, we can use machine learning to identify patterns that predict outcomes using cross-validation to develop classifiers. And unfortunately, I don't have time to get into that. So, you know, I've mentioned there's many, many different resources for these topics. The final thing I need to say is that we really need to be able to ask, was the study designed with the proper controls to make the inferences that people are concluding? Brain imaging using fMRI is correlational. So if somebody really wants to say that the DLPFC causes changes in placebo activity, you need to disrupt the DLPFC and see using things like TMS and see if there are effects. And finally, we have to talk about reverse inference. Simply seeing activation in a region does not mean that a function was occurring. Lastly, simply seeing two different maps doesn't mean that those regions differed significantly. So for instance, here one group has instructions, one group does not. And even though we see activation here in the striatum and the amygdala in the instructed group, when we directly compare them, there's no difference. So researchers need to make sure that when there are conclusions about laterality or group differences, we do not attribute those two differences without actually testing that statistically. So in summary, I hope I've convinced you the brain doesn't simply light up. There are a lot of decisions that go into making, to getting to the point where we see results. These are made from start to finish, and there's many researcher degrees of freedom that people need to read critically and ask about. I want people to continue to consult these resources, including our primer, and things like pre-registration in open science, as well as collaboration with experts in the field, will continue to improve pain imaging. So again, thank you to Masi and Tim and Neil and Petra and the Pain Research Forum.
Wonderful. Thank you so much, Lauren, and congratulations for covering a lot of material very effectively. I mean, that is quite an endeavor to dive into so many different aspects of fMRI, but I think you've sort of covered it so nicely. And maybe I just want to start off with one of the questions coming in from the audience on physiological noise. So you didn't talk about this for time reasons, but it was on one of your charts, and it's very nicely laid out in your paper as well in the primer, that obviously there are sources from what we call physiological noise, like the heartbeat, like CSF pulsations, like breathing, that contaminate our signal. Yes. And rather than asking you how we control for that, because there are tools and you describe them in the primer to do that, I think there's a very, and it comes out a little bit in the question from the from that individual in the audience. I think what is really interesting about this question is that, well, autonomic arousal is so taught, or whatever the to pain itself. So wondering if we actually perfectly control for physiological noise, what are we left with when we talk about pain? Do you want to comment on that?
I would be happy to. I think that's a really exciting question because we're trying to figure that out specifically in my lab right now. So we are revising a paper trying to test, do we come up with better, more specific predictors of pain in terms of testing whether we can see brain responses that are uniquely related to pain and not salience and not temperature, but the subjective experience of pain? Can we remove arousal as a proxy for salience and see anything left over. And so I think this is kind of an empirical question. There is research recently from Vitaly Napadal's lab suggesting that you can better classify patients if you integrate brain activity and physiological arousal. So it's contributing some variance above and beyond brain activity, but whether that variance is perfectly related to pain, I think we have to see. So I think this is like a really interesting question and an empirical one that we're trying to work on right now.
Excellent, thank you. And maybe before I move on to the next question from the audience, I would like to throw something in from my side and maybe just point everybody to that. What I thought was really great, that when you showed the first blobs, you showed them actually on raw functional images.
Yeah.
And you see that very, very rarely. And I hope that people realize that the blobs we saw later on the so-called anatomical images look quite different, because now they're on a high resolution, spatially high resolution image. And this is typically what we see in papers. And then that's why I appreciated you showing the raw images, because that gives a better sense, I think, from the noisy environment the data are actually coming from. So question to you, Lauren, do you think we should actually publish raw EPI images in our brain imaging papers? So for the audience, sorry, just EPI images being the typical functional image.
I guess, so I mean, I love thinking about this. I think it is an opportunity to talk more about websites like OpenPain and OpenFMRI, where we can kind of submit our data to repositories and do open science and share our data. So maybe not in the paper, but I think if we said that, you have to make a movie and upload it or something in addition to uploading your statistical results, or every time you put in raw data into a repository, if it generated the images, that kind of requires people to be taking a look at their raw data. Because sometimes if you're, so the reason that I kind of showed that package from AFNI is because I first learned how to do fMRI with AFNI in 2003 when there were no default assumptions. And so we had to make decisions about how to build our pipelines by looking at effects on the level of a single voxel. And as I moved to other software that has default assumptions, it becomes really easy to just click the buttons and get some kind of statistical map in the end, but not know that half of the brain was missing, or there are spikes, there are all these kinds of things. So if there are ways for us to maybe not publish, but at least host openly the quality of the data, I think that would be great.
Yeah, make available. Great, thank you. I would like to move on to another question from the audience. So there's a question with respect to treatment effect. So it's a specific neuropathic pain question, but I think we can widen it to, do you think brain fMRI can actually be useful to look at some mechanisms of treatment if we scan patients before and after their treatment?
I think that you have to design the study carefully, just like any outcome measure. So for instance, with the treatment effect, there's going to be changes in pain, changes in the pharmacology, maybe changes. There are many, many changes that might coincide with the treatment for any type of intervention. And so as long as you have proper control groups, or you've measured those different kinds of outcomes and you can control for those, then I think you could use brain imaging as a way to get insight on the treatment effect. There are also ways to do this depending on the type of intervention. People use pharmacological fMRI, so you can look at how changes in brain concentration of a drug actually affect brain responses. And so I think there are many, many ways to use fMRI as a tool to measure the effects or mechanisms of treatment, but it varies based on how people set up the experiment, which is why I decided to try to talk about considerations in general so that people recognize that these are all decisions.
Yeah, I realize that. And as you said a couple of times, I think that's so important to point out. It is a primer, and you really need to, every step needs to be carefully considered if you want to be doing that yourself. But maybe just because there's another question with respect to analgesia, so about the time frame we can expect to see something in the brain. Maybe I just take that as an opportunity to point out, because that's something I hear often being wrongly assumed, that if you see an effect of, let's say, an analgesic in a brain fMRI scan, it ought to be a brain effect. And of course, it's very important to keep in mind that it could be an effect anywhere along the neuraxis, right? So let's say we give a nerve block losing a local anesthetic, hence there will be less afferent input into the brain. Of course, that would be reflected in the fMRI scan of the brain, but it does not mean that the nerve block was actually effective in the brain. So I think that if that's okay for me as the moderator, but I think that's a very important point that often gets mistaken, actually.
Yeah. Exactly. So it depends on what your question is, how you design your study.
Yeah.
But of course, you could use imaging to say, how did that change the response to pain, right? Like, so
right, exactly. You just don't know necessarily where that response change happened, but since the brain ought to be intrinsically, intricately related to the perception of pain, that is, one could argue, where it matters in the end, right?
Yes.
Okay, super. Maybe we have time for one more question, I believe. So another, not that, I think, straightforward one, but I think I will pose it nevertheless because it's a very interesting one. So do you think, Lorraine, that fMRI will be better suited to understand certain types of pain conditions as compared to others? Or, the sort of related question, will the specific pain condition dictate which imaging method we should be using?
I mean, yeah, I certainly think that there are certain types of, again, like if you need to, I think there are some studies, so maybe that same Navidad paper I was referring to measured people pre and post exacerbating their pain using like specific movements to exacerbate pain and saw differences in brain activity. But you couldn't actually look at what the movement itself does to the brain response. So I think that if you extrapolate from that, you can think of lots of populations that might not be able to be studied in the MRI scanner. And so I think that really is a serious constraint. We do have other tools like EEG and MEG and other types of methods that might be more conducive to other different patient populations. Again, there's limitations with studying kids in the scanner. But also, you know, I think the type of stimuli, even if we're talking about acute pain, is limited by using fMRI. So we have to kind of triangulate both fMRI studies and behavioral studies and using other types of methods.
Excellent. Yeah. Thank you very much, Lorraine. I couldn't agree more. So we are at the end of our time. So Lorraine, are there any brief final comments you would like to make on the topic?
I'm really excited that so many people tuned in. It was a fantastic opportunity writing this with Tim and Mossy, so I definitely want to acknowledge them. My collaborators who've kind of worked with me, bang our heads against the wall sometimes on how to model things properly, my lab, and NIH and the Pain Research Forum. I am on Twitter, so if people have you know, follow-up questions, you can find me at Lauren Atlas. And you know, I just hope people kind of continue. I hope I didn't scare you away from it, but instead, you know, help people sort of read papers critically or get involved in the field and just learn that there are decisions to be made all the way through from start to finish. So thank you.
Thank you, Lorraine. So with this, I would like to hand over to Neil.
Thanks, Petra. So as we wrap up here, on behalf of PRF, I'd like to thank Lauren for her really fantastic talk today. Thank you so much, Lauren. And I also want to thank Petra for doing a great job of moderating, and also wanted to thank all of your attendees for listening to the webinar and for submitting your questions. And as Lauren said, if you have any follow-up questions, I'm sure she'll be happy to answer. A couple final notes. We'll have a recording of the webinar available later today on PRF. So if you'd like a replay or if you have colleagues who might like like to listen but couldn't attend, you'll be able to access the recording shortly on PRF. And actually, the recording should be available immediately once we're finished here today on the PRF Facebook page. And then also, I just wanted to remind you to save the date of June 17th at noon Eastern for the next webinar. The topic will be neuropathic pain and skin conditions, and will be presented by Margarita Calvo and moderated by Ted Price. And we'll have more information about this webinar posted soon on PRF. So with that said, again, I'd like to thank everybody for attending, and we really hope you enjoyed the webinar. And also one last item, as Lauren mentioned, she's put together some resources for you, and we'll have that up on the PRF webinar page, and we'll also have a follow-up email as well that will have a link to those resources, so you can stay tuned to that. Thanks again, everybody, and hope you enjoyed it, and we look forward to seeing you at the next webinar. Take care.